Cloning is the process of producing genetically identical individuals of an organism. The entire process of cloning is basically extracting the original nucleus from an animal somatic cell, then the DNA extracted from the somatic cell is transferred into an egg cell that had its original nucleus stripped away. This newly formed embryo is then grown inside a surrogate and brought to terms for as long as it can live. There are two common methods of cloning: therapeutic cloning, which is extracting the nucleus of a skin cell and is inserted into a fertilized egg whose nucleus has been removed. The nucleated egg begins to divide repeatedly to form a blastocyst. In reproductive cloning, this newly created embryo is placed back into the uterine environment where it can implant and develop.
The modern day of cloning applications can be found in medical treatments, genetic research, and one that's very important is agricultural industry. In surgery, you are like in the future, we're looking at maybe even cloning certain stem cells that can help human beings who are looking for a genetic matchup or a shift in medicine that can that's called therapeutic cloning, and you can even repair the brain, like damages that happen to the brain, and regenerate broken organs like livers, pancreas, kidneys, etc. Certain effects and disorders like Alzheimer's can be reversed, and all of this is currently being researched. But this can actually bring a new wave to medical treatments and bring a new stage to medicine that can cure using stem cell cloning. And as more research intensifies, we take one closer step to these practices in our medical life.
The agricultural industry looks towards cloning as a marketable strategy to deliver a consistent product. Bananas found in the wild are typically much smaller and contain an abundance of seeds, which make them a lot more difficult to consume as opposed to genetic counterparts, or GMOs, that are often found in grocery stores, which contain fewer seeds and are much larger. These modified bananas often have a lack of viability to grow on their own, which often leads scientists to clone these fruits to sustain the demand from the consumers. The process of epigenetics is also key within the cloning process. Epigenetics is the study of the change in gene expression that results in alterations of the phenotype without completely removing or altering the genotype of the organism. By removing the nucleus from a somatic cell and adding in an oocytoid and adding a surrogate mother, you should be able to make a clone. This recipe, however, does have its flaws, with a very low success rate between 1 to 5%. This is due to an epigenetic barrier that prevents a zygote to bond with the SENT embryo or somatic cell under nuclear transfer.
The current state of knowledge for cloning the stem cell has advanced tremendously, and now we have techniques that is feasible for human cells, but scientists say that the amount of risk is too high. There are 3 different types of cloning, such as gene cloning, reproductive cloning, and therapeutic cloning. Clones live a normal lifespan, but clones like Dolly the sheep only live like 6.5 years, but she lived only 6.5 years because she had progressive lung disease and severe arthritis, so she had to be euthanized. Scientists have been perfecting the cloning procedure since Dolly the sheep was first cloned, so they have been perfecting it and adding new things to it. But cloning is mostly used with farmers to produce healthier and better cattle so they can send them to auction so they can get more money.
Currently, cloning is associated with an increase in animal suffering when compared with production of animals by standard breeding methods. This is due to surgeries performed to obtain oocytes or transfer embryos, pregnancy losses, sickness and death of neonates, low-level abnormalities in surviving young, and possible distress from disease in animals produced as disease models. The moral aspects of cloning has been a hot topic since the concept of cloning was brought to mainstream attention with the first successfully grown mammal in a laboratory, Dolly the sheep. Speculations arose that soon after Dolly was created, scientists would play with the idea of creating an exact clone of a human. This discussion weighs heavy into human welfare and a religious ideology of whether the clone would harbor a soul in suits of that spiritual nature. The lifespan of a clone is also relatively short in comparison to its original counterpart. This shortened lifespan plays into the question of cloning's morality in general.